Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Okay, let's unpack this. Imagine a fatal brain disorder, right? Linked to really low CoQ10 levels that stuff vital for energy. But here's the kicker. The standard CoQ10 supplements, the ones you see everywhere, they just don't work for this. Sounds like a dead end, doesn't it? Well, this deep dive reveals something pretty groundbreaking. Specific intermediate molecules, think tiny head group precursors to CoQ10, seem to bypass that whole problem. They actually get into the brain and, amazingly, rescue a lethal neurological condition. So our mission today is to explore this fascinating recent Nature paper that unveiled this new pathway. We're going to dive into how understanding these tiny, really intricate details of a metabolic pathway can lead to such a massive breakthrough, for treating severe diseases that seemed, untreatable. What does this mean for future medicine? How we solve these complex biological puzzles?
Yeah, it's genuinely a remarkable story in metabolic medicine. Just to give a little background, coenzyme Q10 or CoQ10, it's absolutely critical, like for nearly every cell in your body. Think of it as a key part of the machinery making cellular energy, especially in mitochondria, the cell's powerhouses. And beyond that, it's also a potent antioxidant, protects cells from damage.
So it's fundamental, basically. Not just important, but core to how cells work.
Exactly. And the real challenge comes when CoQ10 levels drop, particularly in the brain. Low brain CoQ10, it's directly linked to some severe conditions, things called primary CoQ10 deficiencies. They can cause lethal brain disorders in babies, infantile encephalopathies, devastating stuff. And it's also implicated in more common things like neurodegenerative diseases, even some aspects of aging.
Okay, so if it's that fundamental and we know people are low, why couldn't we just give them CoQ10? I mean, you said supplements are everywhere. What was the big hurdle, the frustrating part this research tackles?
All right, that's been the sticking point for ages. Despite how important it is and how available it is, just giving CoQ10 supplements, it's been largely ineffective,
Yeah.
especially for the central nervous system. The finished CoQ10 molecule, it just doesn't cross the blood-brain barrier very well. Not enough to get to the levels needed inside the brain. It's a bit like, I don't know, trying to mail a giant package through a tiny mail slot. The package is fine, but it just won't fit?
Ah, okay. So it's less about the brain cells not being able to use it and more that they just couldn't get it delivered properly.
Precisely. And this is where the sort of ingenious opportunity came up, thanks to a relatively recent discovery, this mammalian CoQ10 head group synthesis pathway. Basically, there's an enzyme, HPDL, that makes a molecule called 4-hydroxymandylate, or 4-HMA. And this 4-HMA then gets turned into 4-hydroxybenzoate, 4-HB. These are the key head group precursors. They're like the smaller building blocks for the main CoQ10 molecule. So the hypothesis, the really clever idea, was maybe these smaller, more soluble precursors could diffuse into the brain more easily. And then the brain could, make its own CoQ10 right there on site, where it's needed most.
Wow, that's a totally different angle. Instead of forcing the final product in, give it the raw materials to build it there, how'd they even start testing that? That sounds tricky.
Well, they started with a really powerful tool, an animal model.
Right, the HPDL mouse model. That sounds like the critical first step for this kind of study. Tell us about these mice.
Yeah, this model was specifically engineered, designed to mirror a human condition, a severe, ultra-rare, and sadly lethal mitochondrial encephalopathy called NEDSWMA. So these HPDT mice, they're born lacking that HPDL enzyme, just like the human patients. And what happens is, well, it's quite tragic. They lose weight rapidly, develop really severe neurological issues, spasticity, seizures, and they typically die by postnatal day 15, P15, the paper calls it.
Yeah.
They also have much lower levels of that 4-HMA precursor in their blood, matching the human situation perfectly.
Okay, so a really accurate but grim model. Then came the test, the actual treatment. What did they give these little pups?
So they started giving them oral doses of either 4-HMA or 4-HB. Yep. And they started super early, like between day 3 and day 5 after birth. That early start seemed really important.
And what happened? Did it work?
The results were, well, frankly astonishing. This simple oral treatment completely transformed the outcome. Instead of dying by P15, something like 90 to 100% of these treated HPDIs, they lived to adulthood, basically a normal mouse lifespan.
Whoa. That's incredible for a condition that was lethal within 2 weeks. And you mentioned the paper contrasted this with other things.
Yes, and that's absolutely crucial. They tried standard treatments too, giving regular CoQ9 or CoQ10 itself, even other antioxidants. And none of those improved survival. Not at all. So it really highlights how uniquely effective these specific head group intermediates were.
Okay, so these specific molecules are doing something fundamentally different. But how did they know for sure they were getting into the brain and actually being used to make CoQ10 there?
That's where the elegant biochemistry comes in. They used isotope tracing. Basically, they labeled the carbon atoms in the 4-HMA and 4-HB molecules, like putting tiny trackers on them. And they found that, yes, both of these labeled precursors were effectively incorporated into CoQ9 and CoQ10 right inside the brains of these mice. So it proved two things. One, they crossed the blood-brain barrier, and two, the brain cells used them to make their own CoQ.
Incredible. Okay, so survival is obviously huge. But did it actually help the brain itself? Like
Yeah.
structure, the cells? Did they look healthier?
They absolutely did. The untreated mice, their cerebella, that's a key part of the brain for movement, were much smaller than normal. And inside the cells, the mitochondria, those powerhouses, their structure was all fragmented. And there was widespread cell death, apoptosis, in several brain areas, cerebellum, cortex, striatum, thalamus. Really significant damage.
Yikes. And the 4-HMA treatment, what did that do to the brain structure?
Well, this is where it gets really fascinating. The 4-HMA treatment dramatically improved how things looked under the microscope. It increased the size of mitochondria in the cerebellum. It largely restored the normal structure of the cerebellum itself. It even improved how the dendrites, the sort of receiving branches of Purkinje cells, developed. And those cells are critical. Most importantly, it significantly cut down on that cell death in all those affected brain regions.
Wow, that sounds like it could have massive implications for kids with this condition, right?
Absolutely. These findings really show that this 4-HMA-dependent CoQ production is just vital for normal brain development. It strongly suggests that starting treatment early with these compounds could make a profound difference for children with these HPDL gene variants, potentially give them a much better shot at normal development.
Okay, so the brain structure improved, but did that translate into better function, better behavior in the mice? Because, that's what really matters day to day.
Yes, it did. Functionally, the untreated HPDA mice had problems with energy production in their cerebellar mitochondria specifically, impaired electron flow through complexes I and II, basic energy failure. 4-HMA treatment actually restored this electron flow close to normal levels, and it had a big impact on those Purkinje cells we mentioned.
Right, the Purkinje cells. Remind us why they're so important here.
There are these large neurons in the cerebellum, absolutely central for coordinating movement, balance, motor control. The paper showed that in the untreated mice, these cells were functionally immature. Their electrical properties were abnormal. But the 4-HMA treatment, it didn't just fix these electrical problems, it actually accelerated the maturation of these crucial cells, which is key for developing proper motor skills.
And the human patients, they typically have severe motor issues, right? Spasticity, ataxia. Did the mice show improvement there too?
Yes, quite significantly. In the treated mice, 4-HMA cut down seizure frequency a lot, and it greatly improved their mobility and how regularly they walked their gait. Now, there were still some subtle issues, like their grip strength wasn't fully back to normal, but overall their motor abilities were dramatically better. And it does make you think, if these simple molecules can fix such fundamental cell processes just by patching this one pathway, what other diseases, what other pathways could we maybe fix in a similar way?
That's a really powerful thought. And it didn't stop with mice, did it? This is where it gets really personal, the human impact. There was a case study.
Yes, exactly. A really compelling human case. An 8-year-old child who had rapidly worsening spasticity. And the cause. The very same genetic variants in the HPDL gene that they studied in the mice. Tragically, this child's family had already lost 2 younger siblings in infancy to the same condition. And, critically, standard CoQ10 supplements had been tried for about 6 weeks and had completely failed to help him.
Oh, wow. So a desperate situation, how did they even get approval to try this new approach on a person?
It was done under what's called compassionate use. It required very careful approval from both the FDA and an institutional review board and ethics committee. It really shows the careful ethical steps taken to translate the mouse findings. They decided to treat the patient with 4-HB, one of those precursors.
Okay. And the results. What happened with the child?
The results were, well, nothing short of remarkable, honestly. Objectively, his spasticity scores dropped dramatically. They measured it using standard clinical scales, the modified Ashworth scale, spastic paraplegia rating scale. Big improvements.
Yeah.
But maybe even more compelling were the reports from his family. They saw improved endurance, better balance, way fewer falls, and incredibly, within just a month and a half of starting the treatment, he was able to run again.
Run again. That's genuinely life-changing. Incredible. Was it safe? Any side effects?
Thankfully, yes, the treatment was reported as generally well tolerated. They monitored him closely, of course, but no significant side effects were noted. And if you step back and look at the bigger picture, this one patient's story, it provides incredibly strong evidence that these CoQ10 head group intermediates can safely and effectively change the course of this severe human brain disorder. It's just a powerful example of basic science leading to a real-world solution.
This whole deep dive, it really feels like more than just finding a treatment for one rare disease. It seems like almost a paradigm shift in how we think about metabolic medicine, delivering precursors instead of the final product.
I think that's fair to say, yeah. This idea of metabolic bypass, using these small, soluble intermediates to sort of patch up a broken pathway, it offers a really interesting alternative to things like gene therapy, which can be much more complex. It suggests this head group therapy idea might work for other diseases too, maybe involving other ketones or flavins, similar types of molecules. It could be a template, for treating a whole range of cofactor deficiencies. Some are even calling it the start of synthetic cofactor pharmacology.
So where does this go next? What are the big questions this research leaves us with?
Well, there's several key things. First, that critical window idea from the mice. Remember how early treatment was vital? That really raises questions about newborn screening for humans. Should we be looking for these HPDL deficiencies right at birth? Second, it was interesting that even just partially restoring CoQ levels seemed enough for the rescue in mice. Why is that? Are there other ways the body gets CoQ? Or is the functional threshold maybe lower than we thought? Lots to figure out there.
And what about making the treatment even better?
Exactly. That's definitely a focus for future work. Could prenatal treatment help? Maybe higher doses? Or different formulations to get even more precursor in? Could we reach fully normal CoQ levels? Also, understanding tissue specificity is important. Using more advanced genetic tools to turn off HPDL only in certain tissues or at certain times could tell us exactly where and when HPDL is most critical. And of course, the big one. While the human case is amazing, we absolutely need larger clinical trials to confirm these findings in more patients. That's crucial before this becomes a standard treatment.
This has been fascinating. It really shows the power of digging into the fundamentals, doesn't it? Using a simple, drinkable metabolite to fix a complex genetic problem and literally help someone run again. It's pretty profound.
It really is. This deep dive, I think, highlights how sometimes the answer isn't replacing the final missing piece directly. Sometimes it's about understanding the whole assembly line well enough to feed it a simpler part further upstream. It definitely makes you wonder, what other metabolic diseases out there might be solvable with this kind of elegant synthetic cofactor approach? Where else can we patch the system?
That's a great question to leave us with. What stands out most to you listening to this incredible journey from basic biochemistry to a potential new therapy? Thanks for joining us on the deep dive.